Influenza, old and new threats. So Peter Police, who is the author of this paper, is a big player in influenza research. Obviously, you don't get to write big review Nature papers with a single author unless you are a big player. But his claim to fame is the fact that he was involved in the research that resurrected the 1918 flu, and he was a key player in actually being able to make this knowledge available to the entire world.
Yep, and that was with the ferrets and stuff. I think they're going to talk about that at the end of this paper a bit.
I think that he, some of the stuff was in animals, but what I was thinking more was the sort of the initial characterization, as opposed to
like the molecular characterization.
The molecular characterization and like the identification and so resurrecting the virus was very difficult because what happened is that the influenza virus that had circulated in 1918, thought to have killed 50 million people, it then disappeared, and by the time that we had the molecular techniques available for sequencing, was long gone from the population. And I mentioned this previously, that in order to find an active version of the virus that could actually be genetically characterized, scientists had to go and find a place where it was frozen and preserved. And so they found this, I think that it was an Inuit community in Alaska, in this place called Brevik Mission. And they got permission to dig up the mass grave. They isolated the lung tissue from a young woman who had died of influenza and who was buried there. They brought that back to Bethesda, Maryland, where they were doing the work. They were able to characterize it, sequence it, and then they were able to create synthetic constructs of the historical virus and then test the individual pieces one by one in this neutral background to see if they could identify what one piece of the virus was so dangerous. And they discovered that it was not a very straightforward story. Would you agree with that, Dr. DeLay?
About the discovery?
No, about why the virus was so dangerous.
Yeah, so that's what the second paper is going to be all about, and there's a lot of speculation about that. Yeah, well, let's go. They have a hypothesis, and I kind of alluded to one already. And actually, we're kind of finding out some of the similar questions with regard to COVID, because it's like, well, what determines whether it's really bad for you and may not necessarily have anything to do with the virus itself, although of course there's more dangerous variants than others, but it has a lot to do with the condition of the person that's being infected, particularly age, and such other mundanities. But yeah, they're still trying to piece that together. Like I said, while you were setting up the equipment, the case fatality rate for that initial strain didn't make sense, because when they reinfected animals with it after they had brought it back to life, once they'd, what did you call it? Resurrecting. So biblical. Yeah. Once they'd resurrected this virus, it didn't make sense, right? They weren't able to produce this huge disparity compared to a normal flu virus in animals.
They were in some cases.
That was the main problem. They weren't able to get it nearly as deadly as the actual original strain. Yeah, that's what this whole second paper is about.
Interesting.
Yeah.
All right. We shall get there. We shall look at the detail. It's hard with a science paper because there's so much text that if you miss a single sentence or you pay attention to two different sentences, you can get different results based off of your read. And I actually noticed that he contradicted himself somewhere halfway through the paper, where he seemed to indicate the fact that they had figured out why it was so dangerous, and then in the next paragraph he was like, we have no idea.
Well, here's this line from the abstract. Although more virulent, they're talking about the original strain, although more virulent than contemporary H1N1 epidemic viruses in animal models.
Oh, you're looking at let's deal with that later.
The increased virulence is not sufficient to have been the sole cause of the high mortality rates recorded in humans during the epidemic. So something else is going on.
Right, exactly.
Back to this paper.
Something to note is that as we start talking about all of this research when it comes to influenza or it comes to COVID or Ebola or whatever else, is that in order to understand what is happening inside of these viruses and inside of these viral processes, very often science has to turn to something that is called gain of function research. And gain of function research is, I would just say straight up, very controversial, and there is not a clear decision as to whether or not it is worth doing. Because what gain of function research does is inside of highly controlled laboratory spaces. We're talking BSL-4, which is biosafety level 4.
These are like the guys in the spacesuits, you know.
Yeah,
with
this
the
is
breathers
positive
and
pressure bunny suits. For context, tuberculosis is only a BSL-3 laboratory. And so that at that point you have your bunny suit and you have a positive pressure respirator, but your suit isn't positive pressure. When you get to BSL-4, you're literally in these space suits working inside of a highly regulated containment environment. And the reason that they have to work like that is because they're taking viruses that are maybe middling danger and they're creating very, very dangerous viruses so they can study what it is that makes them dangerous. And on one level, yes, you have figured out what makes it dangerous, but on the other level, you've basically created a bioweapon that if it gets out into the world, you have no way of stopping it because you might not have developed treatments yet. You might not know how to track it. It might move faster than you can get out in front of it. And so it's...
And that information, if it becomes public, can be used as a weapon too, which is this whole other weird level of it.
Right, because
if we know what makes it dangerous, then the bad guys know what makes it dangerous too. And then, you know.
And genetic engineering of a virus is not particularly difficult. You can do it, you could do it at home if you really wanted to.
It's like first day grad school molecular biology. Like if you want to get a gene into a bacteria or a cell line or human cells, anything, you're going to use a viral vector to do that. So manipulating viruses is like kindergarten level molecular biology for modern scientists. So yeah, it's stuff you could like do in your bathtub probably if you had a nice bathtub. And that's really scary.
And it's really interesting that police, who's involved in this kind of gain of function research, writing in this huge, big name journal, in his abstract is pointing out that, hey, this is scary enough that you shouldn't stop us from working on it because that's going to stifle scientific progress. And it's not necessarily relevant to the influenza story. It's just very relevant to this story of gain of function research and the role that police has in it and how it is that we study viruses. Because there is the question whether or not COVID came from a laboratory. And if it did, it would have come from one of these gain of function research programs. And that makes it quite the doozy of a question.
Yeah, it's so funny that it's his sticking point and that it's the last line of the paper too, is something about this risk benefit calculation because, you know, restricting these gain of function studies and making these really secure labs is going to dissuade scientists from wanting to do this work is his argument. And like, and the leaks will never happen anyways. And it's like so interesting because it's like, this was written in 2004. I think this paper is pretty old. Mm-hmm. But, you know, that is an unanswered question is depending on who you talk to, which medical expert you talk to.
Indeed.
So maybe it actually is a bigger problem than we thought it was in 2004.
This is definitely true. So we begin with a brief overview of the genome of influenza viruses. 8 single-stranded RNA segments. The viral particle has 2 major glycoproteins, which is this hemagglutinin and neuraminidase. And then it goes on to say that there are 15 different types of I can't say this word. I've only ever read it. In my mind, I'm like, hemagglutinin. But I'm not exactly sure if that's correct. But there's 15 different of the HA units, and there are 9 different of these N units. And so they can circulate in the population and become recombined in order to create different types of viruses.
So this is already a very key difference here. You have 8 strands of RNA in this virus versus COVID, where you have 1. I think that's really interesting right out of the gate. It seems like it's already easier to swap out pieces with this virus.
With the human circulating viruses, there are 3 main subtypes of these hemagglutinins, and there's only 2 subtypes of the neuraminidase, which are frequently found in the human population. But if you go deeper into this, you discover that there are, I think, up to 20 different varieties of each type in circulation. I guess this says 15, but in recent years, I've heard that there's more. And most of the strains that are found are in birds. So you've probably heard of places that we'll call minks. We've talked about, I think a place had to get rid of hamsters. Sometimes you'll hear about big populations of birds that have to be culled. And this is because there's this fear that when you have this epidemic that runs through the birds, that it's going to get into humans. And our experience with influenza and Ebola and even HIV are the drivers for this fear, where we know that zoonotic origins are quite frightening. Then we start to get into the nature of the pandemic, right? Which is different from endemic in that it appears suddenly and spreads around the entire world, and the dynamics of the spread are very different. Where with something that's endemic, it lives in the population at some background rate, and it appears in the population periodically, but the pandemic point of this transmission is when it's basically on fire. Would you agree with that?
Yep. Pan means everywhere.
Mm-hmm.
Although the second paper we're going to read makes some interesting arguments about the timing and the appearance of the first strain, and sort of argues that it kind of came from everywhere all at once. But that might just have to do with the not a very good tracking system.
Yeah, that sounds a little like magic. So this pandemic that happened in 1918 was very contagious, killed a lot of people, was caused by
40 million people in less than a year.
Which is wild. I mean, that's an upper estimate, but it over the course of 3 years, and it's pretty intense. I mean, we're at 5 million now.
It says 40 million in less than a year right here, anyways.
I don't know about that.
That's what the paper says. Take it as you will. Why are you skeptical?
Because I just remember researching it, and there were 3 waves of the pandemic, where it was basically like wave A, wave B was the really dangerous one, and then wave C was the least dangerous one, and the total death toll was the sum of all 3 waves.
I think that last figure you showed during the introduction was 55 million overall.
It was 40 to 50. This is, perhaps we're getting into the weeds here. So this virus comes in 1918, it fades into the background. Then we come to 1957, where all of a sudden there's a new influenza virus with 2 new surface proteins. So you suddenly have an H2 subtype and an N2 subtype. And these viruses are not particularly homologous to the virus that was circulating to that date. So you have 2/3 amino sequence identity with this previous subtype, and the N2 subtype has an identity of only 37%, which means that there was basically a new virus that arrived. The population was immunologically naive, and so they were pretty susceptible to the sorts of immune catastrophes that happen when you haven't seen a virus before. So they think that in this 1957 pandemic, 70,000 people died in the United States. More died around the world, though I don't think that they were keeping very good records in the 50s. And so this was kind of the clue for people that new viruses were going to start showing up. There's another thing that happens 11 years later in 1968, which is the fact that, again, you have these differences in the surface markers. You have it spread throughout the population. You get this high level of morbidity and mortality. And they think that in excess of 30,000 people were killed in the United States. And only 2 of these genes in this virus were changed. PB1 is the polymerase. And so you had a slightly different polymerase, which is on the inside, which shouldn't really affect innate immunity or even acquired immunity that much, but it was one of the only ways in which it was changed, and so it's kind of thought to be a causative factor in this. But again, you see this huge change. The hemagglutinins differed by more than 60% of their amino acids, which means that that is another huge jump that suddenly appears out of nowhere. And we talk about variants a lot, right? Variant shows up and it grows into the population. And if you have a variant that shows up, it grows in the population, and the population is more or less able to deal with it, it's not that dangerous. It's only when you get a variant that is hugely different, that is different to a degree that it appears in a population that is immunologically naive to it, that you start to get problems.
They also mentioned that the neuroamidase remained relatively unchanged in that H3N2 variant, which is what they ascribed the decrease in deaths for that wave in 68 to. It's kind of interesting. So some parts of it can change, but not all of it.
Or when you have a part that changes and a part that remains the same, your memory immunity is still functional for protecting you. So if you have your N2 subunit hasn't changed that much, then you can survive when you see it again. Yeah, then there was this other H1N1 strain in 1977, but it wasn't really as big of a deal.
They chalked it up to a failed vaccine, actually. Kind of interesting. So they basically immunized a bunch of military recruits with live H1N1, and it didn't work out, and it spread throughout the population to some extent.
It's a very strange throwaway line in a paper where you're just like, well, we did that one. Sorry about that, guys.
Yeah. So
unfortunately, it has been circulating ever since.
Yeah, they were actually, we'll get into this in the second paper a bit more, but they were actually trying out all kinds of crazy vaccinations at the time of the first pandemic. They thought that the virus might have been, they didn't know viruses existed, first of all. So they thought they were chasing a bacteria. And there was a guy, I forget his name, Pfeiffer, I think, who was going around trying to sell this vaccine. And a lot of people were taking it, but it turned out to not have actually any viruses in it that they knew of, since they didn't have a way of actually screening for viruses. So people were just sticking all this weird stuff in their blood at the time. I mean, it's tempting to, yeah.
To be fair, that could help, because somewhere we talk about the fact that if you immunize people against the bacterial things that cause the secondary infections.
That's what I was going to say. It seems to have actually worked out for some population, at least as reported by the people who were selling the things at the time. So,
right. So figure 2 basically covers all of these different variants that are circulating in the population. We have this H1N1 variant that they think is circulating consistently, constantly through the 1950s, at which point we get H2N2. That hangs out for a little while, and then we get H3N2. Then at the same time that we have H3N2, we also have this H1N1, which is related to the original flu strain. And the fact that there is this gap here, to me, is unlikely. I think that it probably continued to circulate all the way through.
I think that's just the vaccine-induced one.
Yeah, this is the vaccine-induced one, but this H1N1, genetically speaking, has never fully gone away. It still is detectable in surveillance of around the world.
Is that so?
I believe so, yeah. And there's also this sort of H2N2, which stops and then it shows up again. And I think that what they're trying to say with this paper is the fact that you basically have this exchange between the surface factors as these viruses circulate through the population. And so you don't have a consistent form of the virus that's there. Every single year, there's a different combination.
You know, there's another way to draw this picture, though, too, that when you take out all the question marks, you don't really see the same strain like popping into existence all of a sudden. All of the ones where it does, other than this vaccine-induced one, are sort of questionable. And so it's not clear whether it's really recycling its epitopes like it appears to be. It might just be, like you said, a holdover from previously or exposure from like a manufactured version.
Mm-hmm.
And they really struggle with that because they're trying to, it's a moving target when they're trying to design vaccines for the flu for the future. They want to find something that isn't changing that they can immunize against, right? Because if they could predict it and have it on hand, ready to go, as soon as one of these breaks out, boom, they already got the vaccine ready to go. And it's not entirely clear that that's possible yet. But this figure makes it seem hopeful, perhaps more hopeful than it actually is.
They go on to basically make the point that there is an interplay in terms of virulence that has to do with both the virus and the host. In an immunologically naive host, virulence is mostly determined by the virus, but even in that situation, virulence is a complex phenomenon. There are many viral genes that can contribute to pathogenicity, and in most instances, virulence is a multigenic trait, which is to say that there is not one single aspect of the virus which is responsible for its damage. You have to have multiple factors that play all together. Then he kind of starts to go into how it is that they figured this out, right? So they look at the reconstructed HA and NA genes of the 1918 virus, and they put it into the background of another neutral virus, and the resulting strain is highly virulent in mice. And this kind of makes sense because that means that it has both the surface markers of the virulent strain and also the mechanism for getting into cells that the virulent strain has. But if you take the HA and NA genes that are currently circulating in the influenza virus, now the less damaging virus, you don't see the same sort of virulence. So already we're starting to tell the story of, okay, so these surface factors contribute to the virulence of the virus that was circulating in 1918.
And so the virulence change, if I recall correctly, is something on the order of like 20x.
Yeah, it's wild.
But the death rate was something on the order of like 1,000x. That's what I think. That's where the contradiction is, not necessarily in the like vector, but in the magnitude, something like that. We'll get into that in the next paper. So yeah, there are definitely molecular signatures that are aiding the virulence, but it's unclear if they're entirely responsible for that massive death rate that we saw.
And this is a pretty slow process, right? So if you, and this is the gain of function that we're talking about, right? You take a regular virus that doesn't have any of these aspects, and then you put these two surface markers into it. You're creating something that can be transmitted more easily through the population. And now you've created something that wasn't there before. And you run the risk of causing great damage if it gets out. Because viruses in their natural state have kind of a balance of aspects that are going to kill the host, aspects that are going to protect the host. And when you engineer it, you engineer stupidly. You don't know what it is that you need to keep in order to prevent it from going off the rails. And oftentimes you don't even want to keep it because you're trying to look to see how to make it as damaging as possible in order to understand its damaging aspects. They show that, if you, depending on which things you move into mice, you get different results as well, right? So if you take 5 of the 1918 genes, you also get highly virulent strains. If you transfer just one of the genes, you actually get a less virulent strain. So it's not a very clear picture. All right, so moving on to sort of where do you want to go from here?
I just want to make sure we have enough time for the second paper too.
Oh, this was really interesting when I read it, which is the fact that so they do these seroepidemiological studies in rural populations where people live with more contact with animals, and they discover that this narrative that they have, which is that there's only a few of the strains that are capable of transferring between humans and animals, is incomplete because you see lots and lots of people that have these H4 to 815 subtypes that they've been exposed to, which is to say that our relatively narrow understanding of how the virus transits through a biosphere is constrained by the way that we live our lives in different countries. So you come to a place like the United States and you look at the way that people live in New York City and you're like, well, the virus can't get from chickens to people. And you're like, but nobody's running into chickens. Obviously, nobody keeps chickens in their apartments. They don't live with farm animals. And if you go to a place where people live with farm animals and they encounter completely different species all of the time, you're going to get a different perspective. And so anytime that we read anything about COVID, it's worth considering the same condition applies there as well.
They briefly mentioned the idea that transmission could be in some part responsible for virulence, even if the case fatality rate is not particularly substantial. They mentioned this 97 strain was highly transmissible.
Mm-hmm. So
they also mentioned, I think this is kind of what you're talking about. Did you already say this? There's a high prevalence of these other strains in the population, despite the fact that they're not necessarily causing disease. Did you get that?
Mm-hmm, mm-hmm, mm-hmm.
So again, viruses are everywhere. We're often infected with them and don't know it. And as long as people aren't dropping dead, it usually stays that way.
I think that this part of the paper is actually quite important too, which is the fact that it's possible that infections of human with these avian influenza strains has been going on for decades, but we haven't been looking, and so we haven't seen them. And that's also what I'm always trying to say about COVID, which is the fact that we've never looked this closely at a virus before. Even these gain of function processes with the flu are nowhere near as much detail as we've given to COVID. And so we have to be careful about these sort of zoomed out conclusions that we make without considering that just observing changes the way that we see the world.
Yeah, this the author actually has, the more I think about it, he has a pretty radical perspective on a lot of these issues. It's kind of fascinating. This line. It may be possible that infections of humans by avian influenza have been ongoing for decades, and it's only the reporting. Did you paraphrase it, or did you? Okay, well, I just wanted to read it word for word. It's just fascinating to come out and say that. So yeah, these have always been there. Perhaps we're just looking really carefully at them. He recommends two remediations. One, that we continue to look at them more carefully so we have a better idea of what's going on. And two, that we stockpile antiviral drugs and develop new vaccines. Now, these are interesting strategies. Stockpiling drugs is an interesting strategy because drugs seem to go bad at some point, which is a real concern. And the vaccines seem to be problematic because the antigens are shifting continually. And so he goes through some different strategies for dealing with that.
He also is surprisingly pessimistic about whether or not the treatments are actually effective against the virus, right? So he's basically, look, there's a couple of different treatments that you can use, and I think that we should go to figure 4 to talk about this.
Okay.
Which is that there's the process of the virus getting into the cell and then fusing and releasing its contents. There is the process by which the virus buds onto the surface of the cell, and then there's the process of actually releasing the virus from the surface of the cell. And there's different pieces that are required for all aspects of this. And he's like, you can use treatments that attack the various steps, but there's problems with them. For example, you get the emergence of drug-resistant variants, which seems kind of counterintuitive because we usually talk about that in terms of bacteria, but viruses also can develop the ability to evade treatments. And so if you have molecular targets that are affecting specific proteins, and those proteins don't have a structural constraint, then they can easily mutate into a new form that is no longer susceptible.
Yeah, it's not even like they're just mutating into a new form. It's just you're just selecting for the ones that can get by that. You know, this what's this? The neuroamidase inhibitors. You know, you can easily imagine that all of the viruses that are susceptible to that just stay stuck on the cell and are degraded, and the few that weren't, that had some minor shape change variation, slipped right by, and all of a sudden, boom, you've just selected for a new variant. And fighting evolution is it's a real pickle.
Right, and he suggests something which are these silencing RNAs. And silencing RNAs are, at the time that he was writing this paper, they were a brand new technology that people had really only just discovered. Normally, RNA was considered to be this nucleic acid that was involved only in the production of proteins, and then people started discovering that there were these little teeny tiny strands of RNA that were somehow functional inside of cells. And what they discovered was that they're called silencing RNAs. The silencing RNA is able to bind to pieces of nucleic acids, DNA or RNA, and then target them for destruction. And so what that does is that basically allows you to stop the replication of a virus, because if you have a silencing RNA that can target the viral nucleic acid, then that will just be digested by the cell's machinery for digesting whatever's being targeted by the silencing RNAs. But it hasn't really proven itself to be a super efficient technology because there's a lot of off-target effects. And so the things that are available to us for actually really effectively dealing with viruses in 2004, not particularly effective. They're driving the evolution of drug-resistant variants. He says something really interesting where if you treat these viruses with these neuraminidase inhibitors, you find that 20% of the children that are treated with them develop the drug-resistant strains, but that and I may have seen this somewhere else, that the drug resistance doesn't seem to make the virus more dangerous. So he's like, at the very least, we're not driving the evolution of more dangerous variants. Is there anything else to say about this in terms of...
No, I think that's right. I realized we skipped a little figure here about the age-related stuff, but let's just save it for the next paper.
Mm-hmm. I think that it's something to notice that the segment on treatments is so short. And this is the case too. So I went into the literature right now and I started looking at, for next week we're doing treatments of viruses, or whenever it is that we meet, because you guys have spring break in there somewhere. We're looking at COVID treatments, and to this day, we don't have very good ways of treating viruses. We didn't in 2004, we don't now. The emphasis largely is on vaccines. Vaccines are the key to controlling a pandemic. And this is a little... You seem to have a lot to say about this section, Dr. Boulay.
Well, that's a very bold statement that vaccines are the key to controlling a pandemic. That's certainly true with some pandemics, and it's been wildly successful with measles and smallpox. And we've actually completely eradicated some of these diseases, which were frequent pandemics across the globe and killing millions of people, which has been phenomenal. So certainly vaccines can control pandemics. These respiratory viruses are a nightmare because their antigens shift in a way that is unprecedented in the viral world otherwise. And so the key to developing an effective vaccine is going to be in getting ahead of the virus and understanding how it's mutating and how to prevent those mutations some sort of before they arise. And it's not clear that we're capable of doing that. It may be possible in the future. Hopefully it will be. But like Dr. Benry pointed out, when you start anticipating how the virus is going to change, you run the risk of creating viruses that are much worse than the one that you were dealing with in the first place.
Mm.
And yeah, it's a real, it's not only an ethical dilemma, but it's a technological conundrum as well. And how do you get out in front of something that's changing so quickly? But when you're in a situation where all of your treatments, keep trying to impress this upon you, your treatments, these treatments have the potential to select for more virulent viruses. Especially, I think what's so unique about these viruses is that they're just transmitted in the, for lack of a better word, in the air, right? They can be solubilized in the vapor in the room, This makes it very quick to transmit them, unlike a lot of other diseases that require direct contact, um, intimate contact, uh, open wounds, ingestion of material, something that's a little bit more involved. But these things can just jump ship really quickly. So, what do the authors have to say about this specifically?
Do you want to just look at figure 5? Because that's the vaccine figure.
Yeah, go to it. Let me just check out the
Yeah,
text
sure.
real quick, make sure there's not something I want to say.
So he makes the case that, look, these viruses are dangerous, they spread through the population, they are poorly controlled with treatments, the treatments are far from where they should be, so we should turn to vaccines. And the vaccines, he basically has a model of how this works, which is the fact that you use these sort of reverse genetic platforms to be able to generate the influenza vaccine strains. You have 2 surface glycoprotein genes, and then you have your donor strain genes, which is the fact that these are the other genes that are produced by the influenza virus, right? We talked at the very beginning that there's 8 pieces of the genome. And then you combine them into a single viral particle that you can then use to inoculate the population. You can use specific gene products that will change the way that the virus is stored. You can change the virulence of the virus. You can very quickly and easily tune for a specific strain as the season changes. And so he's basically making the case that he's like, look, you have a platform here where you could very, very easily switch out any one of these pieces in order to match it to whatever's circulating in the environment at any given moment. And this is the sort of the public health platform right now, which is that, okay, the way that we're going to deal with this is vaccines. But what we're starting to see is, this is really interesting. Jaden sent me a video which was put together by Vox. It was an explainer that was assembled a year ago, roughly. It was in February of 2021, just as the vaccines were coming out. And there is this testimony from the people who were developing the vaccines, who were like, hey, check this out. They're 95% effective, and they prevent transmission. And it's really shocking because now we sit on the back end of a year of real-time study of what these vaccines are actually capable of doing. And we show that they are definitely effective in terms of preventing death, but they're not necessarily effective at preventing transmission. They're not even necessarily effective at preventing infection. They're moderately all right with preventing severity of infection, but we see that we return to baseline something like 12 weeks after the last vaccine, and so the window in which they're effective is very small. And so it'd be curious to see if Police was writing this now, after a year of data, if he would be able to comment on that and be able to sort of to propose some model that extends the lifetime of the vaccine, knowing how mutagenic and how easily escapable these variants are. Because the same problems that he talks about in terms of these molecular inhibitors, where, oh, well, the molecular inhibitors drive evolution, these same problems arrive with vaccine development. And it's kind of sad that after almost 25 years, we're in the same boat when it comes to figuring out how to actually deal with a respiratory virus, which is the fact that we don't have a good solution yet.
Yeah, it was a bit maddening for me, being someone who knew sort of the history of influenza vaccine development and was aware of the problems with it, to hear a lot of the politicians a year or two ago saying, well, we'll just get this vaccine and everything will be over. Because I'm hearing that and I'm like, that's crazy. Like, we've never been able to do that because these things shift so quickly. The best thing that we could ever hope for is doing these really custom tailored reverse engineered vaccines and delivering them in small batches to at-risk populations, like healthcare workers, or in this case, the
Immunocompromised people, old people,
right.
people
So,
with pre-existing
there's an
conditions.
intelligent way to look at this, but telling the population that we're just going to knock it out with a vaccine is, it's just, there's no reason to believe that's even possible with our technology the way it is today. I mean, the paper was written in 2004. We clearly haven't really made, I don't know, measurable progress on even the flu since then, let alone the coronavirus. So, while I do think that the vaccines could be the key to controlling the pandemic, they probably aren't going to make sense in these huge batch rollouts because they need to be updated so frequently and so carefully, and you have to have this very tight monitoring and reproduction and shuffling of the genes, and you really have to just stay on top of it at all times and stay out ahead of it. And honestly, this strategy seems a little like, what's the word? It's a little Neolithic. Like, we're gonna have to come up with something better than this. And he does mention in here that we're searching for this kind of universal epitope that would work against all of these, but that's sort of elusive at the moment too.
One thing that I have seen is that vaccines are being developed that attempt to target the non-exposed part of the spike protein, where you have the stock of the spike protein, and this is the case for these the hemagglutinins. If you can target the stock rather than the part on the surface that's exposed, you have a slightly higher risk of creating a vaccine that isn't just going to disappear with the next variant that arrives. This is a thing that we learned with antibiotics, and we learned it the hard way. I did my PhD in antibiotics and antibiotic resistance, and one of the things that is really a fact of life at this point that is underappreciated is that there was this golden window of time where humans discovered all of the antibiotics that we have. And then what we did is we just used all of them all the time in all situations possible.
Yeah, you can still find them in half of the cleaning products at the grocery store.
Right. They're slightly different now. We don't have penicillin in the cleaning products in the grocery store. We have new ones that we've engineered over the course of the last 10 years or so. But the reason that we had to go and we had to engineer these is because we used these very, very powerful substances across the board without any consideration for the biological underpinnings of what we were doing. And any time that you take a biological entity, whether that is a bacterium or a virus, and what you do is you apply a treatment to it, it evolves so quickly. Its generation times are on the order of hours.
Especially when you apply it to the whole population at once.
You're driving evolution in massive ways. And that's what happens here.
That's the subtext of this paper, which is kind of fascinating. And I feel like it's a really, really unpopular opinion that, I mean, understandably, from the public health standpoint, there's a huge drive to get everybody to get vaccinated. But there is a subtext in these articles, which is like, hey, these are really powerful surgical weapons that perhaps should be used that way. Jury's still out, obviously. But I think we should jump over to the other paper.
Yes.
Should we take a fiver?
I think we probably should. Everybody's been sitting for a while.
Yeah. All right, everybody get up, shake it out, go to the bathroom, and we'll be back here at 50 maybe?
Yeah, let's do 50.
All right, we'll be back here at 50. 2 minutes late, folks.
Both Dr. DeLay and I have a really nasty case of poison oak.
We do. We were scouting to shoot a little section of this movie that we're working on. And we were crawling around by these cliffs, and though you cannot see the leaves on the trees, the poison oak is out there.
No, it's bad. We were
We were nursing our wounds.
we were having an itch session.
All right. Let's go to this other paper.
Right.
Number 2. Door number 2.
This one starts right out of the gate with a fight. The influenza epidemic of 1918 represented the greatest failure of medical science in the 20th century.
Yeah. And they're like, again, I quoted this next sentence, or the third sentence, in our introduction. But essentially, this idea that despite the fact that we can tinker with uh, different molecules and approximate a more virulent virus, it doesn't seem to be effective enough to caused these high mortality rates. I said 1,000 times before. I did the math again in the middle of that last paper, and it was actually 100 times more. So the death rate's about 100 times more than the virulence increase with the news from the original strain. Still a huge discrepancy. So let's look at why they thought this was the case.
Yeah, so there's this idea of this original antigenic sin, which is so dramatic, but we'll talk about that a little bit because there was a very unusual pattern of deaths during the pandemic that the other paper kind of mentioned, but we will go into more detail here. What's interesting to me, which is not necessarily related to the contents of this immunologically speaking, is that people are really obsessed with this question of where did it come from? Why do you think that's so important?
Yeah, that's a hard question to answer. I think because they don't understand what it is that causes the change in the first place. Why does it appear, right? If these are, one predominant theory is that viruses emerge from zoonotic reservoirs. And so if we know where it came from Farmer Joe's pig pen over there. And Farmer Joe seems to be leaking a lot of viruses, then we can clamp down on it. Something like that. I think there's something inherent in the human psyche that wants to seek the source of these dilemmas.
That might be true, yeah.
So yeah, they're going to get into that. There's a number of discrepancies in where it actually came from in the first place. Well, it does matter too. Like in the case of that reappearance of that H1N1 in the 70s, right? If it's really interesting. If the H1N1 actually does rear its head again for natural reasons, that's a very different matter than if you have it being reintroduced by synthetic means. Because one points to biological phenomena that is cyclic, and one points to something that's constantly on the move, and we're going to need different treatment and containment strategies depending on which one of those paradigms turns out to be the case.
Mm-hmm. Right out of the gate, we're dealing with the fact that the World War I was a tremendously damaging experience for the world. The death toll was how are you looking at this right now?
Yeah.
World War 21 death toll. It's not quite what we're not quite there yet. So they think that there were 40 million people who died from the war, and then there was another 40 million people who died of the Spanish flu afterwards. That's about the same number of people that died in combat in World War II, and there didn't seem to be an epidemic that happened around then. So it's sort of, in some way, if you combine the disease and pestilence aspect with the war aspect, World War I and World War II have equivalent death tolls, which I'd never really thought about before. But this was also the birth of modern medicine. We are at the era just past Pasteur. Pasteur was doing his experiments in the late 1800s, I believe. And so we have germ theory is emerging. People are starting to understand that there are these agents that can be communicated between different people. And these agents are going to be able to cause disease. But the problem is that even though we know about these agents, we don't have treatments for them yet because antibiotics have not been developed yet in World War I. So one of the worst complications of a viral infection in the respiratory system is a secondary bacterial infection. And that is basically any kind of viral pneumonia, Streptococcus, Pseudomonas, Staph aureus. You can develop pretty significant internal infections that are the direct result of having the viral infection, though the viral infection is not actually what's actually going to kill you. So it's a very tumultuous time in medical history and human history. And then on top of this, you have the emergence of this new super deadly strain. of influenza.
Then on top of that, you have for the first time, widespread use of chemical warfare. And a lot of that, a lot of those chemicals that were used back then have essentially been taken off of the table. Everybody agreed they were just so heinous that no country was going to use them, which is an interesting agreement, sort of like nuclear weapons. Some countries obviously bring them out from time to time. Let's hope we don't see a lot of that anytime soon. But you can imagine that the cumulative effects of even trace exposures to those elements could weaken the immune system, could weaken the epithelial linings of your lungs. Remember, all these guys, these young men, were stuck in these little trenches on these battlefields en masse, like millions of people stuffed down in these trenches. A lot of these gases were designed to go to be heavy. They sink down to the lowest level. It's just hard to really grasp the damage that is not encompassed in the death rate of soldiers who got shot and died. We're talking about damage that no one will ever be able to uncover, that lasted throughout the next several decades afterwards.
Mm-hmm.
And interestingly, the average age of World War I soldiers is about 24 and a quarter years old, and the highest case fatality rate for the epidemic was for the pandemic was 28 years old. So that's kind of tantalizing too.
When scientists compared, I don't necessarily have anything to say about the age range. My first thought was that it's hard to tell median versus mode, right? So if the median age is 24, what is the modal age? The modal age,
the mean age. Yeah, I agree with you though. I looked for median, but I couldn't find it. But I found mean. This is mean.
Well, mode is going to be the most common age, right?
Yeah, and it's not entirely clear. Right. I see what you're saying.
Right, so it's like mean, median, and mode. These are averages, but they don't necessarily tell you the same thing. An average gives you the direct,
this one says
direct
the average
weight.
Is 30, so who knows?
Right, so who knows? Anyways, I think that looking at historical numbers and estimates makes it pretty difficult, which is why looking at these papers about the influenza virus from 1918 is interesting, because they've actually done molecular studies. And so they showed that if you compare the reconstructed virus from 1957 and 1968 with the reconstructed virus from 1918, that you have a worse... so first of all, they found a remarkable similarity with virus antigen present in the respiratory tree from the bronchi to the bronchioles, which means that in terms of the presence of antigen, this 1918 virus doesn't seem to be particularly different. In the matter of damage, they find that there's the secondary aspect of damage that is leading to a lot of the problems with the 1918 case, which is the fact that you have severe infection of the bronchi and then the bronchioles and then the alveoli. And the alveoli are the point of gas exchange, because the bronchi and the bronchioles are basically these hard tubes, and they have to be able to expand and contract, but they don't have gas exchange happening in them. That's the alveoli. And so your problems arise when you get down and you start to damage these alveoli, and you get secondary bacterial infections, you have edema, you have pneumonia, you have death. So
that's one hypothesis.
Again, they focus on this hemagglutinin because you have this really important aspect of proteolytic cleavage that has to happen, and there's a motif that is necessary for it to be able to be virulent in both animal and avian tissues. If you get rid of the motif, then it's no longer infectious. And this is the similar sort of thing that we see in COVID with the spike protein. If you mutate the spike protein into a form that cannot bind to ACE2, you lose the ability of the virus to be as damaging to the human body as it would be otherwise. And this is a motif that pops up over and over and over again. You have areas on viruses that are the ones that are key for infectivity, that are the key for virulence, and then you have other areas that if you change them or affect them, they don't do that much at all. there's a bunch of stuff about,
A lot of this overlaps with what we went through in the last paper,
and
so I was thinking we kind of graze over that.
do you have something that you specifically wanted to look at?
I just don't want to repeat what we already talked about, and they do to some extent here. Maybe move over to the unique clinical elements. This is where we're going to talk about the age ranges, which we skipped from the last paper, so maybe that makes sense.
Mm-hmm. This is original antigenic sin. Okay, excellent. So, original antigenic sin. This is based around the idea of this odd age range in this pandemic. So they basically say, so in every single pandemic of influenza that we see, this is the case, strangely enough, not the case for COVID, but it is the case for all influenza pandemics. Influenza is dangerous for the very, very young, and it is dangerous for the very, very old. You get a U-shaped mortality curve where you're going to get a lot of deaths on one side and on the other side, but not a ton in the middle. But if you look at what happened during the 1918 pandemic, there's not a figure for this in here. Let's switch over. If you look at what happened in the 1918 pandemic, it was different. Most of the fatalities
were between the ages of 20 and 40 years.
You have this normal U-shaped curve, but it is interrupted by this W in the middle. And the explanation that's given for this is original antigenic sin. And original antigenic sin is basically this, that you have an adaptive immune system. The adaptive immune system is the one that produces the antibodies, and the antibodies are specific to the antigens that you encounter. It is a learned system. We've talked about this. B cells, T cells, antibodies, neutralization, etc. In a normal, strong immune response, you have antibodies that react to the predominant surface markers. So if you've only seen a virus once, you will react to whatever is most prevalent on its surface. And it is possible to have multiple antigens present on the surface of a virus because you have virions, you can have multiple viruses that infect the same cell. As their nucleic acids are being processed, they can be shuffled together. And so you can end up with these kind of chimeric viruses where instead of just having one antigen present, you have more than one antigen present on the surface. And normally there's a dominant antigen, which is why when you look at textbooks and you see the picture of the virion, you're like, oh, well, it looks the same. It has all the same proteins. And it's like, kind of. Most of the proteins are the same. And so from that, you would develop this strong immunity. But when you look at original antigenic sin, you have your patient has already previously encountered this infectious agent with a specific set of antigens. But now the new infectious agent has the inverse surface markers. It has a ton of these surface markers that were present only minorly in the first infectious agent. And so your body basically reacts to these recessive antigens without being able to react to the dominant antigens. And so you never get a sufficiently strong signal to activate the immune response because your body is reacting through memory to these recessive antigens. And so you have weak immunity because it's not the second time that you've seen it. It behaves as if it's the first time.
It's interesting because that's the opposite of how the first paper explains it. So the first paper actually says that what's happening is that in this age group, the drop in mortality afterwards is a result of the fact that the older population had been exposed to the H1 previously in the 1880s, and they had residual immunity as a result of it.
So it's fascinating because scientific terms tend to drift. I'm shocked that original antigenic, does he describe it as original antigenic sin?
Because he's explaining the W phenomena.
So these two papers have a different explanation for the W phenomenon. So if we go back here, what Dr. DeLay is saying is that if you go back to this, what the first paper is saying is that this decrease in deaths is caused by the fact that these people who were above the age of 30 had encountered a virus that had already conferred to them strong immunity. And so they were not seeing the virus for the first time. They were seeing the virus for a second time. And so without this sort of memory process that was in them, this curve would basically have been straight up along the diagonal of the rate that you see between the age of 14 and 30.
I also found a recent paper that suggests a rewrite of the paradigm, which says, although original antigenic sin has been historically depicted as a problematic response, recent data, this is about the flu, have demonstrated that in certain contexts, eliciting original antigenic sin may also be beneficial.
Definitions and understandings change, and this is the thing about science. Somebody looks at you don't have the answer, right? You're looking to see what is happening. And so you have 3 different ideas here, and all of them are held by people in the field. So one idea is the fact that the reason that these people didn't die is because they'd seen the virus before. Then the other paper says, well, the reason that these people died is because they had seen a very similar looking virus before, but it wasn't quite right.
All these may be true, too.
Yeah, exactly.
They could all be kind of working together. They all might be completely wrong, too. My favorite hypothesis is they were all just poisoned from the trenches because they were 28-year-old men. But, who knows?
So basically, this is kind of just another representation of original antigenic sin. You have a virus that you encounter, you see some slightly modified version of the virus, then you see an even more modified version of the virus, and you continue to have stronger and stronger immune responses to the very, very first virus, but your immune response to the third virus that you see is weaker than it is to the first one. And this titer, as it rises in response to the third virus that you encounter, is what short circuits your immune system and causes death because it's not producing the right antibodies. It's producing the antibodies that are not capable of neutralizing the pathogen. That's why I took it.
Yeah, that's a really controversial idea the more I think about it too, because it's actually arguing against the vaccination strategy too, because you might be making antibodies to something that when it mutates down the road will be setting you up for a less effective response.
And this is why we live in a world where, look, if somebody tells you to trust the science, it is usually due to the fact that they have not looked deeply enough into the literature to discover what an absolute morass the science is. That's kind of a depressing thought, right? Because we want to believe in this idea that there's something stable and steady and something that we can grab onto and we can say that this is certain, we understand it, it will never change, and it soothes us in our belief that it will never change. But the reality of the situation is usually very, very different. The reality of the situation is that as you push against the very edges of human knowledge, you are going to find signs that point in all different directions. And the deeper your questions, the less certainty that you will have. And so when we find ourselves in a situation where the entire world is captivated by this question of, what does the science tell us to do? The problem is that you will encounter as many answers to that question as you have people asking it. And it rarely comes down to the fact that there is real, unequivocal consensus at the edges of what is known. And that's the thing that's scary about being in the middle of a pandemic, and people are making decisions based off of science, and we are at a time where it becomes very difficult to talk about unpopular science, such as the original antigenic sin argument being a solid reason for why vaccination would be a bad idea. Because if you then encounter a virus that has a totally different antigen profile, but still has some of the original antigen, you're going to end up in the situation where people won't be able to respond to it.
Maybe.
Maybe.
I'm actually less hopeless about it because I try to always remind myself that we're toddlers scientifically as a civilization. And, we literally came up with antibiotics like 100 years ago. So there's a lot we have to learn, and your generation will be critical in moving that ball down the field even further. Of course, the treatments that we had for disease 300 years ago literally seem like torture chamber events compared to what we have right now. And I'm sure that our treatments today will look like that to people 300 years from now. So science is a work in progress, and it's our job to understand fundamentally, before we even go into the lab and start working, we need to understand what problems need to be solved. And it seems like we have a really hard time as a civilization talking about what problems are out there. We want to believe that we're awesome. And we are awesome, but we're just not perfect. So back to the paper.
You know, I think that we should take a look at the statistics paper.
Yeah?
Yeah, because I mean, there's like some stuff about the ferrets, but
Hold on one second.
this is what I was talking about. So they're talking about old and new, the idea of being able to develop a universal vaccine, which the first attempt at a vaccine for the flu virus was this catarrhal vaccine, which was a vaccine that was catarrh is the phlegm that you cough up from your deep lungs. And the vaccine contained a bunch of bacteria. So it had pneumococcus, streptococcus, and the gram-negative H. influenzae, which I don't know what H stands for here. Formerly bacillus. But these are all bacteria that are commonly isolated from the respiratory tract of sick patients. And so what they basically did was they took the stuff that came out of people's respiratory tracts, they heat treated it, and then they used that as a vaccine. And that actually seemed to decrease death, because what it focused on was not on trying to deal with the virus that they didn't have a good method of actually dealing with. It focused on dealing with the secondary bacterial infections. And so they tested it on 21,000 people. This is also a really interesting story because a lot of these people didn't give, the idea of medical consent was kind of non-existent. They just kind of gave it to stuff. The first...
Mostly military people
mostly
and prisoners.
military men and prisoners, yeah. This was kind of stunning to me. The first smallpox vaccine was developed by this tinkery doctor who identified the bacterium. He figured out how to do it. And he was working off of the old paradigm of variolation. So variolation was this thing that they did before vaccination, which was that you could take a smallpox scab and you could make a cut. So you take a smallpox scab off of somebody who's sick, and then you make a little cut on the arm of somebody who's healthy, and then you take a bandage, and then you literally just bind this smallpox scab onto the body of somebody who's healthy. And that causes an immune response that is attenuated because it is not full smallpox. It's basically mini smallpox. And this doctor, who I can't remember his name, he kind of extrapolated where he decided that it was clear that this was an effective method of preventing smallpox from happening because there must have been some agent in there. And so he figured out how to isolate the agent. He figured out that the best thing to do would be to vaccinate the patient as opposed to just applying it to the skin. And then he tested it on his gardener's son without telling him what he was doing. The son survived. He didn't get smallpox. And from then on, we've basically had vaccination. But the history of vaccination is kind of a weird history where medical history in general is a weird history. When you really start looking at how medicines are developed and the technologies along social paths that they travel, you get into this really weird environment where you understand on one hand that it's useful and it brings good to the world. And then on the other hand, you have to ask yourself the question of, well, would we have achieved this good if we had maintained a different moral center around what it meant to obtain consent and to conduct these trials according to our modern moral sentiments? And the answer is probably we would have gone a lot slower and not developed as much knowledge as we have now about biology, which is a hard thing to live with when I, pop aspirins to deal with poison oak. The point is that this catarrhal vaccine, which was bacterial in nature, actually really did help reduce the risk of death. So in severe and complicated cases, only 8% of those who had received the vaccine died versus 27% in the group who didn't get the vaccine. So obviously, this was a pretty significant advance. And he even mentions here, such trials are no longer regarded as ethical, as they often use the principal investigator, which personally I think is fun. I think that if you're a scientist and you want to test stuff on yourself, have at it. Where it gets to be really weird is that you find that people are testing medical treatments on their friends, their colleagues, and serving personnel, oftentimes who have no idea what's about to happen to them.
I actually went back and reread this section on the antigenic sin, and these authors are not disagreeing with the other authors. So what they actually were saying is that the younger patients that were dying in that 20 to 40 group, they're suspecting that they were exposed to an H3N8 virus, which provided them with ineffective immunity, while the older populations were exposed to the original N1, or H1N1 virus.
Mm-hmm.
And they are calling this phenomena antigenic sin.
Yeah, so it's being exposed to something
So
that doesn't
exactly. It is, so that makes sense.
Yeah, exactly.
It's all totally speculative, though. It's not like they have a sample of the H1N1 or this hypothetical H3N8.
This is true.
Why don't we open the floor up for discussion?
Sure. One last thing.
Marks on this?
Yes, one which is that the best strategy that we have right now for developing any kind of long-term efficacy vaccine is looking at these non-variable regions inside of the viral structure. If we can figure that out, then we stand the chance of actually being able to develop something that's effective. If we can't, then we're just going to be in a closed loop for a really long time, I think.
Yeah, it's often said that science changes by revolutions, so it's highly likely that a technology we can't even imagine right now will completely rewrite the paradigm on how we go about treating viruses and viral infections. I wouldn't be surprised if that has to do more with the way that we teach our immune cells how to behave than it does with actually going after the virus itself. But time will tell, my friends.
If this is something that you are curious about or interested in, it is a field that needs brilliant people that are thinking outside the box and considering these questions in new ways. And there are great rewards for anyone who can find the answer to this, because the humans need your help.
So true.